So thanks for joining us this evening. I'm Becca Young, I'm a PhD student at Cardiff University, and I'm going to talk to you not so much about my project but more about parasites in general and why I find them so interesting. So first of all, what is a parasite? So it's an organism that spends at least part of its life cycle living on or in another organism, which is described as the host. And specifically, to be classed as a parasite, they have to have a negative impact on the host. They come in all shapes and sizes, from microparasites like malaria that live within the blood cells, or macroparasites like some of the larger parasitic worms I'm going to show you today. And they are disgusting, but they're also very interesting, and I'm hoping that I can convince some of you of that this evening. So if you're a bit of a nerd like me and you're in Tokyo, you may be interested to visit this museum, so the Meguro Parasitological Museum in Tokyo. It was established in 1953 and is home to 60,000 specimens of over 1,500 different species. And here are some examples of some of the things there. They've got this really cool book with taxonomical drawings and stuff as well. They've got 20,000 papers and 6,000 books there as well. So it's a treasure trove. So you can see here this one sort of in the middle that looks like a heart. It is a heart. So that is a dog heart which is infected with Diarrhoefilaria immitis, or dog heartworm. Next to that there's some examples of the life cycle of human hookworm. So they've got some samples there. Also shows you the life cycle, which you can't really see here, but it's great in the museum because it's really good for education as well. And this large one over here on the right is the stomach of a minke whale, which is infected with Anisakis simplex, so another roundworm. And as you can see, like I said, they can get pretty big. So another example of a large one is in the museum. It is hailed as the longest tapeworm recovered from a human. I did try and find verification of this because there's sort of reports of other longer ones being found, but they tend to be reports from unverified media sources. So I'm not going to say definitively, but this is definitely very long. So it's 8.8 metres long, or 28 feet. It was recovered from the intestine of a 40-year-old male, and it's believed to have come from some trout sushi. So here you can see it sort of spiralling back and forth because it was that long. This zoomed in picture shows the individual segments, or proglottids. So each one of these contains eggs. So the proglottids are shed from the end of the tapeworm, and then they're released in the feces of the host. And this allows a continuation of the life cycle because it's basically how this parasite continues to spread, by shedding its eggs. So yeah, I said about they have their life cycles. Generally, parasites have a specific host that they need to get into to complete their life cycle. And some of them have very simple life cycles, but some of them are very complex and will go through lots of different hosts. And they need to make sure they end up where they want to be. And there's some very clever ways of parasites have of doing this. And this is one of the things I find most interesting about them, is their ability to manipulate their host. So for example, this Dicrocelium dendriticum, it has part of its life cycle in a snail, then it is shed from the snail and gets into ants. But its final life cycle it needs to be in is cows and sheep. That's where it completes its life cycle. So how's it going to make sure that this ant ends up getting it to a cow or a sheep? So they basically move into the ant's brain, move to a cluster of nerves, and basically get involved in some mind control of the ants. So they cause them to climb up to the top of grass and then just bite on and not let go. So they just stay up there, so they're more likely to be eaten by the sheep and the cows. And what makes this the extra level of interestingness in this is it's temperature controlled as well. So they register the drop in temperature overnight. This is when the ants will go up so that they're there in the morning when the cows and the sheep are grazing. But if they don't get eaten during that time, the temperature starts to raise. They'll sort of relax their mind control, so to speak, and let the ant let go and come down so that it doesn't just dry out in the hot sun. Another parasite that commonly affects snails, its definitive host is a bird. So how is it going to make the bird eat this snail? So the parasite grows mainly in the liver of the snail, then it sends out these branches that burrow into the tentacles, and there they form what's called brood sacs, which are full of larvae. They come in these bright colours. You can see here, it's not normal looking snail tentacles, they're very brightly coloured. They also will pulsate as the larvae are moving around. Basically, it makes them look like caterpillars, which is obviously more attractive for a bird to eat than snails. And another thing they do is they change the behaviour. So nocturnal snails are more likely to be out during the day when they're going to get eaten by birds. This is an example of a cestode, so Schistocephalus solidus, that lives in stickleback. So these small fish are an intermediate host of this parasite, but the final definitive host is a bird. So again, this parasite needs to make sure this fish gets eaten by a bird. And the way it does that is by diminishing the fish's fear response, basically. So normally, if a shadow passes over the water, that's a sign of maybe a bird predator flying over. So the fish's reflex is just to hide, go into undergrowth, go into where it is harder for them to be caught by the birds. But this parasite diminishes that response, so that fish are more likely to stay in the open water and be more easily caught by the definitive host. And humans are not immune to this. This is an example of guinea worm. So this is a waterborne parasite. So the way that this affects this parasite is transmitted to the host through drinking water containing the larvae. So when it's ready to reproduce, this parasite will come to the skin and form painful boils which burn. And the human response to try and relieve this pain is to cool them by putting them in water, which is exactly what the parasite is aiming for, because it will allow it to complete that life cycle as a waterborne parasite. So this is something that is on the decline. So through educating people to teach them that, you know, this is going to increase the spread of it if you do put it in water, and also just the supply of more clean drinking water means that guinea worm is on the decline, and we're quite close to being able to eradicate this parasite. But not all worms are terrible. So obviously there are plenty of bad diseases caused by worms, but there's also some good things that they can do. So the worms trial is at University of Nottingham. It's in clinical trials at the moment, and it's worms for immune regulation in multiple sclerosis. So for a bit of background, you may well have heard of the hygiene hypothesis. So this doesn't just relate to excessive cleaning lowering our exposure to bacteria. Also, parasitic infections are rare in the Western world, and this is possibly having a bit of a negative impact on our health overall. So there's two different types of immune response within the body. So there's the Th1 response, which is in response to bacteria, and there's the Th2 response, which responds to parasitic infection. And to some level, these two responses sort of suppress each other. So an overactive Th1 response can result in autoimmune diseases. So many inflammatory diseases are believed to be caused by an overactive Th1 response. And this may be because, due to the lack of parasites in the body, we're not initiating this Th2 response, and therefore that's not suppressing the Th1, and it's kind of allowing it to run a bit wild within the body. One example of an inflammatory disease caused by this is multiple sclerosis, or MS. And so, yeah, they're trialling controlled infections with human hookworm, which induce an anti-inflammatory immune response. And hopefully this will not only work against the parasite, but also against the inflammation caused by the MS, causing the MS symptoms. So, yeah, it's in trial at the moment, but hopefully we may be able to put some of these worms to use. So finally, I'm going to talk a little bit about my project. There we go. So I am working on the European turtle dove. This is not the most seasonal picture. It's better at Christmas, but they have their fame at Christmas. So on the second day of Christmas, the two turtle doves. But if current trends continue, we won't be getting these turtle doves at Christmas because they're in a severe decline. So they live in the UK and across Europe, and since the 1970s there's been a 98% decline in the UK population. There's lots of contributors to this decline, but one of them is believed to be a parasite called Trichomonas gallinae. So this is a protozoan parasite. It lives in the throat where it causes, in bad infections, it causes these lesions, which if they get too large, they can result in death by either anaphylaxia, so suffocation, or starvation because they just block the throat and the airways. They're common in birds of prey, in doves and pigeons. So it's really common in columbids, which is doves and pigeons, and it is known to cause mortality in turtle doves. It kind of rose to fame a little bit in 2005 and 2006 when it was believed to be a passing from columbids to garden birds, so passerines like greenfinches and chaffinches, and it caused massive declines because it was very, very lethal to these finches. And in Great Britain, the greenfinch breeding population has declined by 66% since the emergence of this disease. So it is often asymptomatic in turtle doves, but as I said, it has been known to produce mortality. So it's something that we need to monitor and to see how infections vary amongst populations, and also if there's different strains of the parasite, so slightly different variants of this parasite, some of which may be more likely to cause mortality, some of which may be more likely to be asymptomatic. So that's one of the things I'm looking at. So to do this, I'm using DNA-based techniques to detect how prevalent it is within a population, because as I said, it's often asymptomatic, so I can't necessarily look at a bird and tell if it's got it. So I take a swab from inside its mouth and throat, which is what I'm doing in this picture, and then incubate it and then test using DNA techniques to see whether it's there or not. So yeah, I'm looking at populations from breeding grounds and wintering grounds. I'm currently analysing the genetic data, so hopefully watch this space and maybe I'll be doing a talk again to let you know how the parasites differ between these different populations. So all that's left to say is thank you all for listening, and I hope that maybe I might have converted some people into being slight fans of parasites.